A very good morning to all the participants in this conference. I'm Dr. Mrs. Elizabeth Margaret, Associate Professor, Department of Botany, St. Anne's College for Women, Hyderabad, India. It's indeed a great privilege for me to be a part of this conference. And at the outset, I would like to really appreciate and congratulate the organizers for organizing such a big event and making it a big success, and also inviting me to be a resource person for this conference. The topic that I have taken for the presentation is a comprehensive study on ethno pharmacological antidiabetic potential of traditional medicinal plants of India. So coming to the beginning of what is diabetes mellitus. Diabetes means, it is the meaning of the word, it's a Greek word which means to pass through. And this word has been introduced by Aratus in the 2nd century. The adjective mellitus means honey, and it has been introduced by John Rollo. Diabetes mellitus is a metabolic disorder wherein human body does not produce or properly use insulin, a hormone that is required to convert sugars, starches, and other food into energy. Diabetes mellitus is characterized by constant high levels of blood glucose or the sugar levels. Coming to what is the estimated growth rate in India? The number of people suffering from diabetes is increasing every year, and the disease now kills more people than AIDS. Till date, 41 million patients were diagnosed with diabetes in India versus 40 million HIV patients all over the world. And if you see, the number will be doubled to 87 million by 2030. Coming to the physiology of the insulin, insulin is secreted by pancreas. Insulin is secreted by two kinds of cells, that is beta cells, glucagon by alpha cells. Pancreas secretes about 40 to 50 units of insulin daily in 2 steps, which is secreted at low level during fasting and secreted at a higher level after eating. An early burst of insulin occurs immediately after 10 minutes of eating, then proceeds with the increasing release as long as hyperglycemic condition is present. Insulin is a small protein consisting of a chain of 21 amino acids linked by 2 disulfide bridges to a B chain of 30 amino acids. You can see in a small diagram where insulin chain A is interlinked by disulfide bond to insulin B. The beta cells have channels in their plasma membrane that serve as glucose detectors. Beta cells secrete insulin in response to the rising levels of circulating glucose. Diabetes is of 4 types: type 1 diabetes, type 2 diabetes, gestational diabetes, and pre-diabetes. Coming to type 1 diabetes, which is called as insulin-dependent diabetes mellitus, IDDM. Type 1 diabetes is also called as immune-mediated or juvenile onset diabetes. It is caused by an autoimmune reaction where the body's defense system destroys the insulin producing beta cells. People with type 1 diabetes produce very little or no insulin. The disease usually occurs in children or young adults. Patients completely depend on the exogenous insulin to control the levels of glucose in their blood. Genetic factors are also believed to be the main reason for it. Type 2 diabetes, which is called as non-insulin dependent diabetes or adult onset diabetes. It accounts for at least 90% of all the cases of diabetes. It is characterized by insulin resistance and relative insulin deficiency. This occurs mainly due to the loss of functional beta cells. Type 2 diabetes is associated with very serious life-seizing complications. Gestational diabetes, which is called as GDM, is a form of diabetes occurring or consisting of high blood glucose levels during pregnancy. It develops in 2 to 4% of pregnancies, generally in the second and the third trimester. GDM usually disappears after pregnancy, but women with GDM and their offsprings are at an increased risk of developing type 2 diabetes later in life. Pre-diabetes. This is a body warning sign to make some health changes. Pre-diabetes is diagnosed when the level of glucose in the blood is higher than the normal limits but not quite high enough to diagnose as type 2 diabetes. It is a symbol of impaired glucose tolerance. What are the risk factors that are involved in the development of the diabetes? Mostly it family history, obesity, hypertension, high triglycerides, low HDL levels, elevated glucose, polycystic ovarian syndrome, vascular disease, history of baby weighing more than 9 pounds, and a sedentary life cycle, and schizophrenia. What are the associated complications of diabetes then? Stroke, retinopathy, kidney failure, heart disease, and amputation of the foot or the leg. If you see the zone-wise report of diabetes in India, it is the western part and the southern part of India where you can see the prevalence of diabetes is more, whereas in northeast and in the northern part it is quite less. Coming to the traditional medicine, the world WHO has listed about 21,000 plants which are used, of which 2,500 plants are in India, of which 150 are used commercially. Because India has a rich heritage of these traditional medicinal plants, India is called as Botanical Garden of the world. Of all this, 400 plants are used for diabetes. The most common drug, metformin, which is used for treating diabetes, is obtained from Gallica officinalis, and the compound that is present is guanidine. And why herbal drugs are to be used? As the victim of diabetes mellitus are increasing in the normal population day by day, there is a strong need for the development of natural remedies, which are free of the problems associated with allopathic treatments and also which are able to fight against all the complications associated with DM. Hence, WHO recommends the further investigation of traditional medicine for the treatment of diabetes. And the herbal drugs are natural in origin, they are effective, they are safe, and they are also affordable and they are mostly accepted. We will just run through what are the common plants that are used by the different parts of India, the people who are present in different regions. If you see northeastern India, you can see the leaves of Acacia consina, they are infused and they are taken orally. The decoction of the root from aconitum is also used. Adathoda vasica, which is a very common plant used for cough syrup, the infusion of the root is used. The whole plant of Acumia facetica is also crushed and mixed with water. The leaves of ale marmelos are boiled in the water and the water is consumed. And then there are a number of plants like albizia, alocasia, aloe vera, alstonia, Annona squamosa, Annona scomacus, Andrographis paniculata, Artocarpus, Arica catechu, Artemisia, Beta vulgaris, which is beetroot. These are the plants that are mostly used for treating in northern eastern India. Coming to eastern India, if you see, the plants, the treatment is the same. Either the leaves are boiled in water or the seeds that are used, or the leaves are made into a paste and then they are used. The common plants that are used are Calotropis gigantea, Cajanus cajan, Cisalpinia pulcherrima, Brassica, Cisalpina cristata, Bougainvillea, Bombax, and the other plants are Alstonia, Ananas squamosa, Coccinia, Costus, Cynodon, Derris sandens, Abrus. These are some of the plants which are used by the people in eastern India. Coming to the treatment done by the South Indian people, there are plants like Betula, Cisalpinia, Caparis, Eugenia, Azadirachta indica, which is the neem, Allium cepa, Eucalyptus globosa, and the plants that are used by the central part of India, Madhya Pradesh, you can see there is a big list like Allium cepa. The common plants are Allium cepa, Allium sativa, Ale marmelos, Acacia fistula, Acacia tora, which is found everywhere, then Hibiscus rosa sinensis. And these are the plants which are commonly used by the people from Madhya Pradesh. What makes these plants medicinally important and why people prefer these plants? The most important thing, the biomolecules that make these plants medicinally important to beat diabetes. What is the mechanism of action that is involved in the herbal remedies? Acacia arabica, it actually initiates the release of insulin. Agrimony eupatoria, it releases insulin, and also insulin like it mimics insulin action, insulin like activity. Allium cepa stimulates the effect on glucose utilization and antioxidant enzyme. Aloe vera, which is a very common plant, stimulates the synthesis and insulin secretion. Artemisia palens, it inhibits the renal glucose absorption. Beta vulgaris, it protects the destruction and regeneration of the beta cells. Azadirachta indica, the neem plant. The glycogenetic effect is due to epinephrine action which is blocked. We will now just run through some of the common validated plants that are used in the traditional medicine to treat diabetes. Coming to the most important plant, Karela, Momodica charantia, it contains an active compound called charancin and momodancin, which is reported to possess pancreatic beta cells, and insulin is released and it fights the problem of insulin resistance. Another plant which is commonly known, Tinospora cordifolia. This plant contains the alkaloids, the isoquinoline alkaloid rich fraction from the stem. It has been reported that it has an insulin mimicking property and insulin releasing effect. Another compound isoquinoline alkaloid called berberin, it has been tested and it is used successfully in experimental and human diabetes. It actually lowers the elevated glucose and has the same effect as metformin. Trigonella phoenicum, which is methi, the seeds are usually used as one of the major constituent in Indian spices. The seeds also improve glucose metabolism and they normalize creatine kinase activity in heart, skeletal muscles, and the liver of the diabetic person. Aloe vera, the whole plant is actually very important. The gel of aloe vera, it possesses significant anti-diabetic cardioprotective activity and it maintains superoxide dismutase and catalase activity. Moringa uliifera, it is actually involved in the stimulation of the beta cells and subsequent secretion of preformed insulin. Acacia arabica, the seeds and the bark, they contain the polyphenols and tannin, which reduces the blood glucose by the release of insulin from pancreatic beta cells. Allium cepa, a common plant which is used, that is onion. The active ingredient, that is allyl propyl disulfide in onion, has antidiabetic property. The hypolipidemic effect of onion is due to its active biomolecule allyl propyl disulfide, which prevents hypercholesteremia that leads to the heart attack. Ale marmelos, which is a common fruit. The fruit, the seed, and the leaf extract, they contain coumarin, which potentiate the insulin secretion from the existing beta cells from pancreas. The leaf contains eugenol, is supposed to possess anti-hyperlipidemic activity. Allium sativa, the garlic, it increases hepatic metabolism and it releases insulin from the beta cells. The neem plant, Azadirachta indica, the bark from the root extract and the leaf extract of Azadirachta have shown antidiabetic activity. It contains a compound called azadirachtin A and nimbolite of neem leaf. The extract that possess hepatoprotective role against the liver toxicity. Costus, which is called as the insulin plant, is rich in protein, iron, and antioxidant components such as ascorbic acid, alpha tocopherol, beta carotene, terpenoids, steroids, and flavonoids, which all have shown significant anti-diabetic activity. Gymnema sylvestris is also another plant which is used to lower blood sugar, reduce the amount of sugar absorbed by the intestine. It lowers the LDL cholesterol and stimulates insulin release in the pancreas. Cynodon dactylon, which is a common grass, the plant contains a number of saponins and they actually block the formation of glucose in the bloodstream. The flavonoids, they suppress the glucose levels and release plasma cholesterol and triglycerides and increase the hepatic glucokinase activity and enhances the insulin release from the pancreas. Sistrum nocturnum, which is called as the night queen, the phytochemicals that are present, they are also used. The flavonoids are also known to regenerate the damaged beta cells in the diabetic patient. Ziziphus is another common plant which is used. The plant contains a number of antioxidants which are actually present in the fruit and they have hepatoprotective nature. Lawsonia inermis is another plant which is also used. The extract of the leaf, they have hypoglycemic action due to pancreatic secretion of the insulin from the beta cells. Aristoloche indica, as the name goes, it is found in India. Aristoloche indica was found to produce good antidiabetic activity in treated mice. It actually reverses the weight loss of the diabetic condition and restores normal blood sugar levels. And it shows significant antioxidant activity. There are a number of products that are available in the market which are made from all these plants. You can see fenugreek, karela, betamelon, dar beta plus, madhumeha tak churna. Now coming to the last part of the lecture, the potential research challenges in future. Although many plant species have been validated for their antidiabetic properties and related complications, a need exists for research in the following areas. Identify the active phytochemical compound which is associated with hyperglycemia and conduct a number of extensive large population clinical studies and most importantly investigate combination dosages of natural plant products and synthetic drugs to determine the optimal combination for cost-effective therapies. And also, one should determine the long-term side effects. And another challenge is determine the exact mechanism behind the hypoglycemic and anti-hyperglycemic activity of the plants. And I hope my presentation has been useful. And if there are any queries, you can contact me. My mail ID, Margaret_NJE, which I have given in the first slide. For any networking or for any collaborations, you can contact me. Thank you very much.